Okay, so this is our fourth session, and I'm going to dive pretty quickly into the steps we're going to run through today because there's a fair amount to cover. Um, so what I'm going to end up going through is at the last point we had reviewed all of the steps in running our level 1 regression models. That's our models that we run for each individual subject. And I'd like to start out today by kind of going through and showing you visually what actually happens in each of our steps. So we'll be taking a look a little bit in AFNI about what happened exactly in our session. And then the majority of the time I'd like to spend running through some basics of QC review. And there's a very nice tool that AFNI has that helps to automate some of the more basic processes. So I'm going to hop first onto the cluster here. So I've gone through our usual setup where we ask for an allocation from our QMaster node and then logged into that node with this flag so that we have Xming forwarding in order to be able to display visually things that are occurring on the cluster up here. The steps that we would take in order to run our script. So let me just bring up our T-shell script here. So this should be fairly familiar, but this is basically our setup where we gave a bunch of variables so that the program could find our files, and then specified everything that we would run through within our regression, what model we were using. So then what you would do after that step, when that's all complete, is you would run this program called run.sh. And basically what this is set up with, just to quickly review, there's a flag for what kind of script it is. And then there's a bunch of information about sending the job to the cluster so that you can have it operate on the script that you set up, and you don't have to have a window up while you're running it. And all this script does, essentially, other than give you information about what type of, or give the cluster information about what type of job you want and how to notify you when it's done, it just ends up running that script that I just showed you. So to set that up, you would just end up typing sbatch, which says to the Slurm workload manager, which is the queue system on the cluster, that we want to run this script. And then what it would, if I push to enter here, what it would do is start the script, and then some hours later, 3 or 4 hours later, it would send you an email in your email saying, hey, the job's done. And since I don't want to spend 4 hours waiting, I set things up so that we could look at an example that was already set up. So if we take a look, our subject data from that script ends up being stored here in this demo subject directory. And then if we look here, there's also a results folder. I'm showing you more than I would ordinarily, just so you can see the structure. And then this is our real results folder, so we end up with here the output of the script, and this is the actual script that it ended up running. And that's just the translation of everything that we typed in AFNI ProcPy into a form that it can just run. And some other time we can go into more depth, but in here is all of the results, and you can tell that there's numerous files here. Basically, what AFNI does is it wants you to be able to access at any time any information that you need in order to basically see each of the steps and evaluate steps if there's problems. So you'll notice that there's a bunch of head and brick files here, and these represent each of the steps that we go through. It's an image of what happened in each of the steps, and each one builds on the other until we're finished with our analysis. The other thing you'll note in here is there's a lot of .1D files. Those are basically text exports that AFNI generates, as well as a couple of graphs that it uses when you're doing quality control review. And then there's a few text files that show what the output ended up looking like. There's also a few commands, and we'll go through these in QC, that it's generated. So in my current color scheme, this could vary, but things highlighted in light neon green here are programs that you can actually run, or scripts that you can run. And one of these scripts is at EpiReview. This lets you look at a video of your raw data, and we'll look at that later. And there's also these at ss_review, and there's basic driver and driver commands. These are also all different parts of the SS review program. The basic is really, really basic information about how your regression went. And review driver is like a semi-automated process that takes you through each of the steps to see if your data looks good. So before we go into those, I wanted to take a quick look here and show you what the steps actually ended up looking like. So now we're going to end up going into a GUI. I'm going to launch AFNI, GUI being graphical user interface, and that's why we needed Xmin, because it'll pop up here. AFNI has an internal sense of humor. This is the day Caesar crossed the Rubicon, apparently. Okay, so this is our AFNI interface, and a session or two ago I showed you guys just a little bit of this. But what the program ends up doing, I'm going to make these a touch bigger, is it looks in whatever the current directory is, and it searches through, and what it's trying to do is find an image file, because it wants to try and make your job easier. So it goes and finds the first anatomical image file that it can locate in the folder, and if for some reason you can't, it can't find an anatomical image, it'll take the first epi or functional image that it can find. And so in this case, what we're looking at, and if you mouse over this, it may not quite show it, but what it's showing is it's showing demo subj, which is the name of our subject number in this case, underscore anat, and it's plus a ridge. You can't quite see it. So this is the T1 anatomical image, the structural image that they take at the beginning of our scans, that shows us the structure of the brain, and it's really high resolution. This is the kind of thing that makes your results look real good when you put them in a published figure. I'll talk a little bit about that later. But this is actually this individual subject's anatomy. And as we were talking about before, in order to get a bunch of different people in a group, a bunch of different subjects together, we have to kind of warp we have to warp our epi images into the same common space so we can talk about them as a whole group. And so the anatomy is the basis that we use to decide how to warp our epi images, because its resolution is much higher, and we're warping them into a template space. And there's basically two common ones. The one we use is the Telleract template, but all that really matters is that the brain images are warped into a common space. So I'm going to click this underlay button, and what this allows us to do is essentially there's two layers in the image. There's the underlay or the background, which is usually anatomy, and you can also use overlay to put images on top of it. Usually it's functional activation. So just to take you guys through the steps here. So one of the first things it does is it ends up going through and stripping the skull out of the anatomy, and then warping it into a standard space. So the first thing it does, and this will look a little weird here, is it actually warps the whole anatomical image and the skull into a weird space. And so you can see some weird warping happens when you look at the outline of the skull. So you can tell this image has been warped, but the goal is to just talk about the brain matter. So it doesn't matter that that's a little weird, because that ends up getting then stripped out. And we have, this is this person's anatomy warped into the template space. So it looks really similar to the template space we're using, but this lets you take a look at what's going on. So essentially we end up getting the information from the anatomy files that get warped. So now you'll see there's these pb00.demosubr01. So in the beginning, all of the pre-processing steps that we performed when we were talking about it last time, these get run on each of the individual runs in our data. So we have 4 runs. So this is PB00, that's the first step in pre-processing. This is who it is, and then it's .R01, that tells us run 1, and then it says .tcat or .tshift. That's the step that it's running through. So for these, I'm not going to show you every run at every step, but I'll just go through the first run. So the thing that you want to note here, you see how this is EP, this is epi, and then it has colon and it says .3D or colon 3D plus T122. What this means is this is a 3D image, so has an XYZ, so you can browse through it. It's not just a flat image. And the T is time. So there's 122 TRs, or basically full scans of the brain contained in this file, and they're ordered in time as you move through. So let me go ahead and switch. So I'm going to switch our underlay to this epi image, and you'll notice a pretty big change. Okay, so this is the hidden truth of neuroimaging here. So this is our real data. This is what the brain actually looks like. So you'll notice a couple of things here. One is it looks all pixelated. It is pixelated, but this is the actual resolution that we can talk about in the brain. So each one of these little squares is a certain volume in the brain. In this case, I believe we're around, give or take, 2 millimeter cubes. It's not exactly that, but that's a good way to think of it in your mind. And so each of the light and dark values here correspond to a different level of signal in the brain. The other thing you'll notice is there's a little bit of information here from the skull. And the trouble with the skull is things can be bright enough. If things aren't aligned right, you could be matching some part of the skull into some part of the brain, which is not so good. So a lot of our pre-processing steps help address that. Primary amongst them is kind of warping the template space. The other thing you'll notice is this image isn't really aligned. So this is the front of the brain and this is the back of the brain, but it's kind of tilted like this. This is because to be comfortable in the scanner, you gotta lie back a little bit like that sometimes. So people aren't perfectly aligned. But you can see that a lot of the oxygenated blood is very clearly in the brain. There's a pretty strong delineation compared to over here, which is not in the brain. So there's some blood in the tissue, but your brain uses a lot of oxygenated blood to run its functions. So the other thing you'll notice is, the other thing you'll notice is right here. So there's some of this is just where the frontal lobe starts, but you'll see there's like a little bit of disruption, particularly as you move like about here. So you notice there's a little bit less signal. This looks a little exaggerated in here. It's not as bad as it looks, but basically the thing that's right below your frontal lobe is your nose. And the thing about your nose is you have air passages in it. The rest of your brain is basically encased, more or less, in liquid, so it has a similar kind of characteristic. But your nasal passages end up, because there's air in there, you end up disrupting the image a little bit. So there's a little bit of distortion. Our pre-processing steps help account for this, but it's important to know that that's an effect in your image. Okay, so what it did right here is it recategorized, it basically concatenated everything. So in our next step, we end up applying our time shift. And this one didn't have a lot of motion, but sometimes what you see is when they're categorized together, you get kind of a striation in the brain. That's because each of the slices happened at a different time. This T shift helps basically align all the slices so that they're occurring at the same time and they're balanced. So those are kind of the basic pre-processing steps. You need to do that no matter what so that your data is properly aligned. The next step is our volume registration. So you'll notice a really big shift here. So let's get in the proper part of the brain here. So the big thing you'll notice is the activation starts to look a lot more brain-like, what we're used to seeing in terms of a templated brain. And that's because it's applied all of those warps that it got from the anatomy. And you can see a little bit of ghosting here from the skull. If you'll remember in that warped image that still had the skull, it kind of bent, you can see that here. So at the volume registration stage, we have a much more clear brain, and it's clear that it's in kind of this templated space. The template space, again, letting us compare subjects to each other. So if you run through the step with a lot of subjects, everybody's kind of in the same common space. And so when we talk about a particular voxel in here, we're talking about a particular area that's been mapped out. We applied a nonlinear warp, so you'll notice too that there's variation. The linear warps just kind of move the brain around to help match it better. Then our next and also very important step is our blur step. So this blur step helps reduce noise It also reduces the specificity of the signal as well. So you'll notice a pretty big shift here. And you didn't forget to put on your glasses. This is basically what happens. So you can see really clearly visually here that when we're talking about this voxel that we're looking at, it's a little more ambiguous, like where that exact point is. However, if we go back a step, the thing you'll notice about is there's a lot of individual variation and things that look a little noisier. Blur helps take care of all of that. So it's a Gaussian blur for the people who want to get technical, but it's got a certain radius that we've assigned as part of our regression. So that helps clean up the data. And then the last step, we won't see too much visual change here. Maybe a slight change in the colors. So this is basically a rescaling of the epi data. So this looks noisier in some ways, but actually what we end up having here is basically everything, including what's in the brain and what's outside the brain, is in the same space across runs. So here we'll have another shift. These all ended up looking the same. While it's subtle visually before and everything looks roughly the same, this step actually really equalizes everything. So when we're looking at areas, we'll be looking within the brain, and this keeps all of our runs comparable. So after this point, everything gets merged into one big long run. And it's on this that all of our analyses are run. So I'll show that in a second. The other thing that's useful to see, so there's a bunch of masks that are generated. We don't end up using it, very many of them, but I just wanted to show you one of these guys. So let me go ahead and bring that up. So this is the extents mask. So what this is, it doesn't look very informative, but what's actually going on here is throughout the brain, there's some areas that are clearly not even part of the scan, and those get excluded. There's also other masks available. So this one is a mask, a pretty restrictive mask of just the areas that we think are in the brain. Now, and then this is a slightly broader mask. It's kind of hard to tell that that's a difference, but these are pretty similar. So the idea with this is if you were analyzing the subject, you could only include those voxels in the brain. But we actually end up using only the extents mask as part of the analysis that it generates. But what happens is in our epi data, only a certain number of voxels are active for our subjects. Stuff outside the brain, there's often some values of 0. And in our analysis approach, what ends up happening is if you have a value of 0, that voxel doesn't exist. And if it doesn't exist in one person, it doesn't exist in your whole sample. So what ends up happening is you don't end up really needing these masks because essentially that process ends up masking out activation across your whole sample. So it's like you kind of take an average and that ends up dropping out all these voxels that aren't really part of the brain across your whole group. Okay. So then the final thing that gets generated, this is our stats file. So what we end up here is an image here where you have hotspots that occur throughout the brain, and you can kind of see a little bit of ghosting of the blur. So this is a little bit of hard to place in context, but brighter things are in our brain image are more active, and darker things are less active. What we usually do to help make much better sense of this is we'll actually end up using our anatomy. I'm going to use this person's final anatomy, but you would usually use the template, but this is so close to the template, I'm not going to worry about it. So you would set that up as your underlay, and then you can set up your stats file as your overlay, what gets shown over. Now, going into the actual analysis is a whole separate topic. But just to kind of show you what ends up happening is you can gradually increase the threshold of what gets counted as real activation based on the statistical output that you get. And when you establish this is being goofy. Hang on a second. If you establish a threshold, you get an idea of this is something in the brain, this is an area that's active, and then you can map where it's active. I don't want to go too far into that because that's a whole separate topic. But the idea is...
Are you changing, like, I can't see that far, but are you changing,
sure.
like, p-values when you scale up
Yeah,
and down
so basically what this scale is our raw, our t-stat
that?
value.
Okay.
And then we have different descriptions of our threshold. We have a p-value and also a q-value. And so that's just basically increasing the water level of the activation, and the islands in here are the places that were active enough to be above that threshold.
Okay.
Yeah. And
Yeah,
it,
so this is
yeah.
not really an activation map. This is a probability that the activation in that area
Yes.
Is different.
was related to whatever your
Yeah.
regression is.
Yes.
Okay.
That's a good
That's
clarification.
a better way to
Yeah.
help understand it.
Okay, so I'm not going to go too much into the analysis, but that's the start and then the end goal of what we're running through. So I'm going to go ahead and close this. So now we're at the step where we would want to do QC review in our pipeline. So we get the email that our job has finished, and we want to go see if what we got was really what we wanted, or if there weren't errors in the analysis set. So what I like to do first is typically look at I like to first use this program, SS Review Driver. And what this program lets us do is go through steps and use the output that we've generated partially in all these like 1D files and stuff to see if what we did makes sense. We'll see this a little bit better. So the first thing it does is it actually will output this file here, and this is actually the same thing that at SS Review Basic produces. So what you get is kind of a report of how things went. So it'll tell you the ID number, and there's a lot of information here. I'm going to highlight the key ones that are useful in evaluating things. It'll tell you here how many types of stimuli you produced. We talked a little bit about our experimental design before, but there's 4 different types of events that we're looking for. It then tells you what data sets it used. This is your nice final double check that you didn't accidentally run the same person or copy the wrong file. So this gives you a last sanity check. Here it reports on the voxels. That's a little bit less informative because that's after it's kind of been resized. So then it has this value, which is our motion limit. If you guys remember back to our regression, this is what gets kept in the analysis is any TR or any full volume scan of the brain. If the motion was more than 1 millimeter, it gets thrown out, and then the subsequent volume also gets thrown out. And that just helps limit high motion scans, because we don't want to match anything where there was a big change in movement, because we might be mapping a different part of the brain to the same space. Our pre-processing steps help mediate that, but they're not magic. So that's why we established that limit. It gives us some reports about motion. That's a little bit hard to really parse. So the other thing I tend to look at is the outlier limit, and that's basically a level of how much signal can spike, and this is a pretty easy, it's a less conservative limit. It lets in more data. So then if you look here, so these lines are really useful. So what these set up is, first it says you the number of TRs, or basically the full scans of the brain that are in each of the 4 runs that we ran through. Then it'll tell you this TRs per run applied. So this is how much of the data got through our motion and outlier limits. And then the second one is how many of those volumes got censored or removed. So in this case, it was 2. Sometimes it's a little easier to look at this next line here, because basically it says what proportion of the volumes were above our limit. In this case, we had really good results. 3 of our runs had nothing above our motion or signal limits, and really there were only 2 TRs in the last, so less than 2%. Okay. So there's some reporting on the degrees of freedom. This doesn't really come up that much, but occasionally there can be some information in errors that will refer to that. Basically, you want to have degrees of freedom left, just like any analysis, like an F-test or a T-test. Okay. This is all good. This can give you a general picture of things. But the one that I use the most actually is this secondary set. So now what it does is it breaks it down. So for our stimuli that we ran, this is how many volumes of each of the four regressors that we put in. This is how many volumes were related to the convolution of their hemodynamic response. So anything from when the event happened, any volumes that were part of that occur over the next few scans. This is how many of those went into each of them.
And the 4 regressor systems, so this is
Yeah.
the go, no-go.
Yeah.
So basically, this is 10 minutes of a kid lying in the scanner, and their job is to do 2 things: either go, don't go. And the 4 regressors are doing that correctly. Go correctly when you're supposed to go. Don't go when you're not supposed to go, or incorrect responses on either one of those. And all of this is just kind of looking at the regression of that regression applied to the data, and how much of that is left and how much of the variance is accounted for. So.
So this next line here, this is the really important one. So this is the number of TRs that were censored per stim. So this tells us how much of the data that we cut out here was actually during something that we cared about in the experiment. So in this case, it's very nice. So we had 2 volumes censored before, but they weren't really associated with any of these other ones. And this is fairly common, because what happens is you're doing your task, and sometimes there's a little gap in between, and you're done, and you go, done. And then in that time, your brain moved a lot. But we don't really care about that too much if it didn't happen during anything we cared about in the task. And so we'll often look at that, or more often, the percentage of TRs that were censored. In this case, we had a really good result. that's because I picked this data because it had low motion so that we could see things nicely. In actual experimental circumstances, you'll get a fair number of TRs censored. The idea is to keep that at a reasonable rate, not have them all be zero. Then the last thing we get is a few different reports about what our signal looked like. There's a whole lot of variety in terms of the statistics that we run through, but TSNR is our total signal to noise ratio on average, and basically what this says is things that we think are signal versus noise on average throughout our whole signal. This is what this value is. Signal in itself is not really all that terribly informative, the actual value, because it's somewhat arbitrary. So this ratio tells us, oh, compared to how much noise we saw relative to the signal that we think related to our task, this is like what we're seeing. So I don't think this is super informative other than to know that when this value is higher, you've got more going on relative to noise. I don't want to talk about these too much. They're a little bit obscure. The last thing that I'm going to touch on is this maximum f-stat. And we'll look at this a little later visually. This is the point in the brain, the peak activation in the whole brain related to our task. At the highest point, it has an F value of 43, which in this case is pretty big. And generally, we tend to look for values that are greater than 20. So this is all in our basic review. This is a good snapshot of how things went, but it's really important to actually look at your data. So I really think this is not sufficient just to look at this. Fortunately, at SS Review, we'll take us through a bunch more steps that will help us out. And it gives you instructions. Hey, take a look at this. Does anything look unreasonable? And then you can click OK when you're finished, and it'll proceed to the next step. Okay, so this is the step where we review our plots. And basically what we're looking at here, there's 3 of these guys. So this is a graph of our signal over time. And this red line is that 0.1 cutoff that we talked about. So here's all the signal in our brain. At this point, there was so much signal that it was above our cutoff limit. And then it gets flagged, and it's a little hard to see, but there's a green line here. It'll be apparent in the other one. So this gives you an idea of how big how generous our cutoff is, because there's a lot of activation down here, and it's clear that this is not really part of the signal, because look how huge it is. We would expect things around 0.01, 0.02 down here, not a big spike. So we're pretty sure this isn't real data.
And that's adjustable, but you want to keep it the same for all the subjects in your study. And sometimes if you have, kids who are wiggling a lot, you want to try to find a good place where you're getting as much data as you can.
Yeah, and that makes a big difference. So this is the graph of our motion, and here you can see the green line really clearly. But this red line is our cutoff, and actually here we didn't really get so much motion throughout. And we got a little signal spike over here, and that's represented in green in this graph. And you'll see, well, hey, while it wasn't above cutoff, there was a lot of motion right at the same moment. And in looking at these two graphs, that's what we're hoping for. Because when you move, you tend to get signal spikes, and they're basically highly associated with each other. And really what you're looking for here is you don't want these graphs to look way like qualitatively different from each other, because that would mean there's something wrong with how your data was brought in. Your motion and your outliers aren't aligned to each other or something, and that's a good flag that maybe you used data from the wrong participant or something, because these things are very highly correlated, so we should see some correspondence. And the last graph here, we see the top in black is outliers, and this corresponds basically. It's like it's essentially a composite of these two values. And I think a norm is related, but it's a slightly different graph. But they're both getting at movement and signal changes here. And then what it gives, just kind of for our information, if we're trying to figure out what altered, is these first three graphs. They're a little hard to see here what the labels are, but these are basically movement in the X, Y, and Z plane that occurred during the task. And then next we have yaw, pitch, and roll, which is basically the rotation that happened within the scan. And these are incorporated into our analysis as regressors to reduce the chance that we're talking about a different voxel than the one we think we're talking about. So what you might expect to see is right around this spike here, there should be some shift in these planes. And that just sort of verifies that, oh, it makes sense there's changes here. You'll notice there's a few alterations here that occur here, 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 and here, essentially kind of breaking things into fours. As you might expect, the reason that's happening is this is the first run, this is the second run, this is the third run, and then the fourth run. So what happens is there tends to be more movement between runs because the task isn't happening. They're within our motion tolerances, but that's why you see that stair-stepped thing. Okay, so these all look good. So then you want to close them, and then we'll run you to the next step. Okay. All right. This is a very important step. So this is our review of alignment. So the whole idea is that we can map the anatomical data and our epi data into the same space and use that data to warp our our epi data using our anatomical correspondence to a template, which is a big mouthful, but basically the idea is the anatomy is our key to warping things right to the template. Now, the problem is if your anatomy and your epi don't correspond well, then you're warping your functional data in a way that might not correspond very well with how it actually is. So then when you're talking about one area, your certainty is less that it's the same spot. Where you're seeing activation might not be quite right. This is the underlying anatomy. And so then we're going to go in and set this overlay. This isn't an actual file exactly. It's a thing. It's a space that's set up so that we can just look at this data quality. So it ends up using the second step, the volume registration here that we looked at a little bit earlier. So this is the volume. Okay. So then what it wants me to do is to essentially flip between these two values, which you can use the U key. I actually end up using the O key. But basically the idea is if you press this, it turns the overlay on and off. You can also do it the other way, but I find it not as clear. So you can also use U to flip between two images. But the whole point here is that gray matter should appear light in the functional data because blood isn't flowing to the other areas. Just as when we look at the anatomical image, light area, it's the reverse. So light areas are the white matter, darker areas are the gray matter. Black is not in the brain, not part of the brain. So when we flip rapidly through, if you kind of blur your eyes and look at this, it should look really close to a brain. And we're really looking at gross anatomy. So if it was wrong, this is our brain, the other brain might be like that. So that's what we're trying to diagnose. Correspondence is never perfect, but we're just trying to see, hey, does this look reasonable? Does this brain look like it's sort of on top of that brain?
Yeah, because the computer matched it.
Yeah.
And most of the time it does a great job, but sometimes something weird happens,
Yeah.
and you can usually see it in this. It's like, whoa, wait a minute, why is
Yeah.
it flipped like that? So.
And I mean, if you typed the letter Q and put it into the same space, this program would find a visual solution to match that up as best as possible.
Right.
So it'll find a solution. It just, the solution might not make any sense. So that's really what we're checking here. Okay. So then it has you close AFNI, and then we move to the next step. Okay. All right. So this is just a view, a set of warnings that could have happened during our analysis. And in this case, nothing went wrong, so nothing showed up. But what this would look like, this first line is a warning in this program, 3D Deconvolve, which is basically your regression program. So this would give you, along with tent here, any warnings about high correlation or any issues that it ran into when it was trying to run your regression. Then it gives you potentially correlation warnings. What this is about is dependencies between your variables of interest. So if events tend to be matched really well with each other, they'll start having correlations in the regression, and you really don't want that because you're looking to separate out each of the events that you're interested in, whether they got it right, wrong, in the right context. So this would tell us if anything was highly correlated. It doesn't necessarily mean that we can't run our data, but it does help identify potential problems. If all of your regressors are highly correlated, your design isn't that great, and you're probably missing results. This is a common reason why we'll go in and apply jitters into our results, because it kind of breaks up the events so that they don't correlate to each other. I'm happy at some point to talk through that if you're interested, but we'll go ahead and move along. So we didn't find any warnings in that results. Usually at this step, we would often find problems, so it's pretty rare that we have issues with this. Then this is more graphs here. So these two are talking about our regression. So ideal sum 1D, this is basically a sum of this. So let me talk about this. So these are our 4 types of events that happened. And if we were looking at single events, it would look like just lines here, like this happened. Like something happened at 6 seconds, there's a line saying this is the event. What this is that convolved with the hemodynamic response. So remember when I was talking about how you might have an event and a couple of TRs might be censored within it due to motion or something? This is how that happens, because it takes time for the blood to move through the tissue and then move out. So in this case, everything that's above 0 here is getting counted in some way as in this regressor. So you notice these blue and green regressors happen more rarely. What happens down here is more elaborate. So these are overlapping events. So our actual graph, we have many events occurring next to each other. And so a little bit at different times gets associated with different events. But what we're interested in is this event as a whole. So we end up with a graph that's counting different periods of activation differently into this regressor. What ideal sum shows us is the sum of all of this. So it gets broken up, but this basically shows throughout the time course of your event how much of all of the images that we recorded are counting towards your experiment, basically. And what you want to see is not a lot of things at 0. You want some things at 0. And you'll notice, again, these correspond to gaps between our runs here. And then what you want to see is a lot of stuff that's above 1 with a lot of spikes and variation. And that just ensures that you're not like saturating your signal. If you have way too many events too close, it just looks flat, and then you can't really delineate what was due to one event or another, because they basically turn into one flat block. So you lose a lot of information about different events. So this is a way to just see, hey, overall, did my experiment look pretty good? So that's how you can get that. So this is also another way to diagnose if in your pilot data, it looks like you can't actually analyze anything, you might rethink your experimental design. But this is mostly a double check. It also tells you if something really weird happened, or if the timing file you put in is wrong in some way. You'll see that pretty quick, because you should see spikes and variation in your data. Okay, so then I close all of those, and then I look at the next plot. So this is a depiction of our f-stat that I referenced a little bit earlier in the max f-stat. That's what we're going to be looking at. So it loads AFNI up again, and here's our anatomical map, and that'll get warped again. And so basically what it did is it pulled our stripped anatomy file, and then it overlaid this full f-stat, which is basically an f value that represents the amount of activation related to all of that, to that ideal sum, basically all the activation that happened in your experiment. And then what it does is it establishes a threshold so that you can see what was counted as part of your experiment. And then it jumps right to that peak. So it notified me that threshold that it said, so anything that's, green or darker in the image, anything that's colored, is an F value above about 6.6. And then you'll notice it jumped to this peak. And so our epi data is in our overlay. So OLay here corresponds at this particular voxel, the F value is 43.02. There isn't a ton to evaluate in here other than you want your peak to be pretty high, basically as high as you can get it, such that your data allows it. And you want this peak to be happening somewhere in the brain. If it's out here, and this was your peak activation, it's okay at this stage to have some random activation outside the brain. That gets accounted for in our thresholding and stuff later, because this is just activation related to anything in the task. But what you really want is you want this peak and hopefully a few other similarly dark red peaks in your brain, in your image, to be in your brain. That means your task has activation in the brain. And there's lots of different models you can try with your data, but if you don't have activation that occurs in the brain, you don't have any results.
Yeah, and I try to like things, we always look at this final sort of have what we used to call it a positive control, sort of something where you expect to see, you look at all of the visual stimuli that occurred in the experiment, the model, you should see, and every single person that you run through should look about like this. You should get a big pop in the visual cortex, or when we're doing auditory stuff, you should get a big, you should see the auditory cortex at a pretty high threshold. And everybody should look about the same. So if somebody doesn't look about like that, you kind of have to go back and sort of with a fine-tooth comb see what was the movement like, how many events were there, so it's a sign that something's different about that person.
Yeah. And in your data, when you move from level 1 to level 2, which we won't really cover level 2 here, but it's really important. You want to be able to see that you are not putting anything strange into that secondary model. Because if you have really weird data, if there's a lot of missing voxels, you're missing whole parts of the brain in your level 2 analysis that you wouldn't get otherwise. So there's a back and forth play when you're designing a study about what threshold you're calling good data and bad data. The important thing is to keep it the same, but also to keep it appropriate to your sample. You don't want to throw out all of your data. You don't want to throw out everybody that moved a lot in the scanner if you're interested in ADHD, for example. So you have to pick something that's appropriate for your data. And there's no gold standards exactly. There's ranges that people think are appropriate, and you just have to rationalize it depending on your data.
Yeah, for example, just real fast.
Yeah, sure.
But when we first did this, the first 10 or so subjects, it just wasn't looking right. And Whit and Michelle and I were just beating our heads, what is wrong? And we ended up figuring out that there was something weird about the timing. We thought our models were great, but there's a difference. They were throwing out scans, or we thought they were throwing out scans, or they weren't. There's something weird like that. But we figured that out only because those positive controls just did not look right. So it's really important to sort of convince yourself the data is good.
Yeah, particularly in the first few subjects or in your pilot data, you really want to make sure that you're getting what you want, because these scans aren't cheap, and you want to make sure that you don't have half your sample have not really usable data. So these steps are particularly important in the beginning of an experiment, and I repeat that mantra, and I try and adhere to that goal as much as I can. Okay, so this is the end of the QC review piece, and we're almost out of time, but I want to show you just really quickly this at Epi review. So I've been extolling the importance of looking at your raw data. One piece that you miss in the SS review is you don't see visually what happened in your experiment. So what Epi review does is it takes a look and it'll show you the real functional data that happened in a course over time. So let me go ahead and start this so it's less abstract here. So it's similar to what we were doing before in that there'll be an OK button and you'll step through them. Right now it'll just load up AFNI, and then it's going to say, hey, this is a pair of data sets. It gives you warnings that these aren't perfectly aligned, and that makes sense because this is our raw data. Okay, we're still loading up here. So what happens over here is what we're looking at, and I can move a little bit in voxels. So what's going on here is this voxels signal series, and I think if it's not doing it for me, I moved it, but oh, there, yeah, here we go. So this little red dot is moving through our signal.
So it's moving through time.
Yeah, it's moving through time. These are the volumes as we're going through. And what you'll see here is, let me make this a little bit bigger so you can actually see it. But you'll notice there's shifting of the light and dark values of the brain here. And this is the blood oxygenation change. See, in a good case like this, what you'll see is not a lot of movement of the brain itself, and you'll see variation and change throughout. This will also show you if there was some really weird thing, or it gives you a hint of visually how the motion looked throughout your experiment. So you can get some characterization. You get some totals in that main view. But with this, you can see what actually happened. So you notice there was like a little jump there. What happened is we reached the end of the motion, or the end of this run, and we jumped back, and that was the change in motion that occurred between runs, which we saw visually earlier.
And this has already been motion corrected, or not? 'Cause usually, if you think of raw data, you see it a little,
What-
it's a little more wiggly.
Yeah, this is the raw data. So
Raw,
this is
that's guys.
yeah,
Very
yes.
cool. Wow.
So what I'm going to do is I'm actually going to, this will take a second to load, but I want to show you, because I know there was a motion spike in there. This will probably take a second. Oh, I messed it up. Okay. Okay. Here we go. Yeah, so what happened is I closed the window and I was just supposed to push enter instead. So this will just take a second. But basically what it ends up doing is it ends up going through each of the 3 sets of runs, or 4 sets of runs in this case. So it ends up showing each piece as you're going. So then at the end of the review, what we would have seen was something very similar to that last little jump between runs. And if you get a subject that's moving a lot, the image does a whole little jig the whole time. And that's really bad. You don't want a lot of like short movement. Things get accounted for much better if you have a movement and it's either abrupt and you move back, or there's a shift and things are pretty static. That helps things work better in the overall results. Unfortunately, the motion that tends to get outliers and things like that occurring in their data where we drop volumes is usually due to that kind of like back and forth kind of wiggle movement. So that's part of why you'll notice so much of the QC review centers on motion. That's because it's really central to ensuring we have good data. Okay, so that's what I wanted to cover in this workshop.